Hello, everyone, and welcome to today's webinar, Optimizing Treatment Options for HER2 positive and ER positive breast cancer using models of endocrine resistance. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit www.thermofisher.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Hilary Stiers, Postdoctoral Fellow, Georgetown University Lombardi Comprehensive Cancer Center. Dr. Hilary Stiers is a postdoctoral fellow training in the Tumor Biology program funded through a T32 Ruth L. Kirschstein National Research Service Award at Georgetown University Lombardi Comprehensive Cancer Center under the mentorship of Dr. Rebecca Riggins. Dr. Stiers received her PhD from Rutgers University in Endocrinology and Animal Biosciences and has always been interested in how steroid hormones, specifically estrogen, influence breast cancer development. For our complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Stiers, you may now begin your presentation.
Hello, and thank you for that introduction. As mentioned, my name is Hilary Stiers. I'm a postdoctoral fellow at Georgetown University's Medical Center in Lombardi Comprehensive Cancer Center. Thank you to GIBCO for inviting me to do this presentation, and thank you to all of you for attending it. First off, I wanted to give you a little bit of background about myself. I did my Bachelor's of Science in Behavioral Neuroscience at Lehigh University, and I followed that up with a PhD in Endocrinology and Animal Biosciences at Rutgers University. And the reason I moved into my PhD in endocrinology was because I was interested in finding a bridge between my neuroscience background and work in cancer research. And so I did my PhD at Rutgers, and then I followed that up, and I'm currently doing my postdoctoral fellowship in tumor biology at Georgetown University. As you can see on the right, my lab is shown there with my group. My Twitter handle is up at the top. One of the things that I've really grown a passion about is tweeting about my research and about breast cancer. And below the picture of my lab, you can see the group of women that I tweet with, and men. There are no men pictures, but there are men who are part of the group of BCSM, which stands for Breast Cancer Social Media. In our lab, we study breast cancer, and as you can see from this map, that breast cancer is the top cancer with an estimated, based off estimated number of new cases for 2018 for females in the vast majority of countries in the world. Over 2 million new diagnoses are made per year worldwide, and it is estimated that there are 522,000 deaths each year. And people don't die because they have a single lump in their breast. Most people die from metastasis. So one important thing to consider is how many people are living with metastatic breast cancer. It's approximately 150,000 people in the United States alone. And 1 in 3 people with early stage breast cancer will develop metastases. So our goal when we study breast cancer is to stop the cancer before it metastasizes using different drugs, as well as slow and reduce the metastases that have occurred. To do this, we subdivide breast cancer into a few different cases. The first subdivision that we do is to look at cancers that express the hormone receptor. So we subdivide the cancers into expressing the hormone receptor or hormone receptor negative, or those that do not express the hormone receptor or hormone receptor positive. When I say the hormone receptor, I'm speaking specifically about estrogen receptor and progesterone receptor. And in this case, estrogen receptor, progesterone receptor are what we call overexpressed, or there's high levels of the protein within these types of cancer. When thinking about how estrogen works in breast cancer, we want to first understand how estrogen signals normally in the cell. So normally, we see testosterone, which is a steroid hormone, as well as which estrogen also is, but testosterone is aromatized using an enzyme called aromatase into estradiol. And estradiol is an estrogen. There are three estrogens: estradiol, estrone, and estriol, which estriol is more often found in pregnancy. But estradiol is the most common and the most potent of the estrogens. And so estradiol is found circulating in the bodies of women who are premenopausal, so their ovaries are still working. That's the main site of estrogen production in a premenopausal woman. And in a postmenopausal woman, they would have local aromatization or local production of estrogen through adipose tissue that's found near their breast or in their breast. And what happens is the estradiol is able to enter, passively diffuse into the cell through the membrane, and then it interacts with the estrogen receptor. Normally, the estrogen receptor is bound to heat shock proteins, or HSPs, which help to stabilize the receptor within the cell. And estrogen, or estradiol, binds to those estrogen receptors and finds another pair of estrogen with estrogen receptor, forming a dimer. And then this dimer enters the nucleus of the cell and is able to bind to specific sites on the DNA called estrogen response elements, or EREs, along with other proteins, including coactivators and the RNA polymerase, leading to transcription of genes. And so that's producing new genes that will ultimately lead to growth and development of the tissue. And the genes that I wanted to point out here, which I'll bring back later, are PGR, progesterone receptor. So estrogen causes production of progesterone receptor, as well as TFS1. And again, this whole process leads to growth and development, so cell division of the cell. So what we use for therapy for estrogen receptor positive breast cancer are to try to target some of these processes to reduce estrogen signaling. The first place is to use an aromatase inhibitor, and there are three examples on the right there, which completely blocks that aromatization of testosterone into estradiol so that there's less estrogen present. Faslodex or fulvestrant, or ICI 1822780, or in the presentation later, I'll be calling it ICI alone without the numbers, is a selective estrogen receptor degrader. And what that means is that it enters the cell and is able to actually degrade the estrogen receptor so it's no longer present. Tamoxifen is a selective estrogen receptor modulator, and what it does is bind in place of estrogen on the estrogen receptor, preventing the estrogen receptor from signaling through the cell. These therapies are largely successful and have saved many lives. However, we have some problems with endocrine therapy. The first is poor adherence, and this article that came out in October of this year demonstrates that patients who are premenopausal with early stage breast cancer, approximately 1/6 of those patients are not adequately adherent to their endocrine therapy, because often these therapies are used for 5 to 10 years. Another thing that happens with endocrine therapy, or these therapies for estrogen receptor, I use those interchangeably, endocrine therapy and estrogen receptor therapy, is a process called endocrine resistance. So what is endocrine resistance? If a patient is diagnosed with estrogen receptor positive breast cancer, that patient will, and the patient needs surgery, they'll often undergo something called neoadjuvant therapy. So in that case, the patient is given one of these endocrine therapies, and then we monitor so that before surgery we can see whether the tumor has responded or not responded to the therapy. One type of endocrine resistance is called de novo resistance, where the patient's tumor does not respond. In other cases, which is more often the case, the patient does respond. However, what happens for the 10 years following is that in some cases, the patient remains disease-free, and about 50% of patients, they experience endocrine resistance, and they'll have what we call relapse. So in that case, the tumor may come back, or the patient may become metastatic at that point. Breast cancer subtypes are subdivided into the hormone receptor positive or negative, but we further subdivide them looking at HER2. So HER2 is another type of receptor that's found on the cell, and in breast cancer, it can be amplified. So that means that there are additional copies of the DNA of this protein within the DNA versus just having it overexpression of the protein. And when we look at HER2 signaling, HER2 is what we call a tyrosine kinase receptor. So the receptor actually sits within the membrane of the cell, allowing for the receptor to interact with different proteins or growth factors that are on the outside of the cell. In the case of HER2, it actually doesn't bind to any specific proteins, but the other members of the HER family, which include EGFR or HER1, HER3, and HER4, actually interact directly with some of these growth factors. And then what happens next is that the two of the HER families will dimerize together. Dimerization is just the process where the two proteins come together and bind to one another. And then this binding allows for a signal to be sent in the form of phosphorylation. So these phosphorylations occur both at the tyrosine kinase receptor or the HER receptor itself, and then also downstream to other proteins that are involved in growth pathways. In this case, leading to proliferation, motility, invasiveness, and other aspects that lead to continued growth and continuation of the tumor. So therapies for HER2 positive breast cancer are shown on this slide, and we can see that Herceptin and Perjeta are both antibodies that block, either in the case of Herceptin or trastuzumab, block the phosphorylation that occurs within the receptors themselves, or in the case of Perjeta or pertuzumab, the dimerization that occurs between HER2 and HER3. Additionally, there's lapatinib, which is a small molecule inhibitor, which actually can enter the cell and block both HER1 and HER2. And finally, we have Kadcyla or TDM1, which is the T in TDM1 stands for trastuzumab or Herceptin. So that's the antibody. And the antibody is actually conjugated to DM1, which is the chemotherapeutic agent. And what happens here is the T on the TDM1, instead of acting as a blocker, acts as something that helps the chemotherapy to find the cells that have HER2 amplification. And so the TDM1 binds to the HER2, and then it's internalized, and the DM1 is then released, causing the cell to die. And so if we go back to look at these breast cancer subtypes, we can see that the vast majority of breast cancers are HR positive, so hormone receptor positive, and that about 5% are HER2 positive and hormone receptor negative. However, hormone receptor negative. However, we have about 10% of all cases that we call triple positive. And triple positive just means that the tumors express both HER2 and ER and or PR. And so we're not really sure how these patients should be treated or how their tumors should be treated, as we have the two separate classes. The HER2 positive treatments work well for HER2 positive, and the HR positive, we have those drugs that work effectively. So what do we do with these patients that are in between? So the therapy guidelines, we use ASCO, which is the American Society for Clinical Oncologists, has guidelines that they release for clinicians to use to determine how to treat patients with these types of cancer. And overall, if the tumor is HER2 positive, it's suggested that the clinicians use HER2 targeted therapies. However, this is the language that's included within the guidelines for both HER2 and ER positive. And as you can see, for HER2, selected patients should also have endocrine therapy or endocrine therapy alone. And for ER positive metastatic patients that also have HER2, they can have HER2 targeted therapy plus an aromatase inhibitor. And that's for those who are not chemotherapy candidates. So it's confusing because the language kind of doesn't really give us a direct answer. But what do the clinical trials show us? So if we look at clinical trials, these are for HER2 amplified cancer. independent of whether they had an ER status. So the inclusion criteria for these studies was to say that the patients had HER2 amplified cancer, and then they were able to analyze the results post-hoc to look back and see how the patients responded, whether there was whether they had they were ER positive or not. So this is looking at pathological complete response, and you can see that the patients who are ER positive do not have as high of a pathological complete response as those who are ER negative. So overall, in HER2 therapies alone, when the patient is HER2 positive and ER negative, they have a better pathological complete response than those who also have ER positive disease. So what about studies that include concurrent endocrine therapy? These studies are newer, and they show that there seems to be an added benefit when you include endocrine therapy with a HER2 targeted therapy. So overall, adding endocrine therapy in HER2 positive, ER positive breast cancer patients improves their response. And these studies looked at how HER2 treatment influenced pathological complete response. But what about the patients that were treated with ER targeted therapy? So kind of the flip side. So we see that in studies where they looked specifically at HER2 and then looked afterwards at ER positive versus negative, ER positive did not fare as well. What about if you're an ER positive patient and you're treated with ER targeting therapies? What happens there? So this is looking at the results from the SOFT and the TEXT trials. So these trials compared in premenopausal women aromatase inhibition with ovarian suppression versus tamoxifen with ovarian suppression. And so SOFT stands for Suppression of Ovarian Function Trial, and TEXT is tamoxifen and exemestane trial. So again, comparing aromatase inhibition and tamoxifen in patients who are premenopausal, and that's why the ovarian suppression is included, because it helps to reduce the amount of estrogen that's present. In this case, they then did, again, this post-hoc analysis where they split up looking at HER2 positive versus HER2 negative. And what you can see is that those patients that were HER2 negative, or sorry, I keep saying HER2 negative. What I mean is that they do not have an amplification. So there's no HER2 amplification. That aromatase inhibition with ovarian suppression, those patients fare better than those with tamoxifen and ovarian suppression, compared to the HER2 positive patients where they fare better with tamoxifen versus an aromatase inhibitor, suggesting that premenopausal breast cancer responds differently to adjuvant therapy. And therefore, estrogen receptor itself may function differently when HER2 is also present. So I've demonstrated some clinical evidence that patients who are double or triple positive that have HER2 amplification and ER positivity have a different biology that's occurring compared to those that are HER2 alone or estrogen receptor positive alone. And so the clinical evidence suggests that the presence of estrogen receptor affects the response to HER2 targeted therapy in these double positive patients, and that the SOFT and TEXT trials, so those clinical trials, suggest that the estrogen receptor biology is distinct in HER2 positive versus HER2 not amplified disease. So what about the preclinical results? So a lot of research has been done looking specifically at HER2 amplified or ER positive, both in cell culture models and in animal models, but there's not as much research that's been done looking at HER2 positive and ER positive disease. One important thing to note is that HER2 and estrogen receptor pathways overlap. So as you can see from here, there are non-genomic responses of estrogen receptor, which I didn't talk about earlier, but they do exist, and they can, the estrogen receptor with estrogen present is able to interact with HER2 or EGFR. Additionally, those downstream proteins of EGFR and HER2 can interact with, enter the nucleus and interact with the nuclear genomic mediated response as well. An interesting phenomenon is that these pathways can switch dependence when one is inhibited. And so I'll give you some examples of that. So HER2 positive cell line, the UACC812s, are HER2 positive, ER negative, and progesterone receptor negative. And so as you can see, if we look at tumor cell growth, in black, we see that vehicle is just the what we use as our control, that there's no effect of ICI or fulvestrant on these cells. As to be expected, there's no estrogen receptor that's present, as is evidenced by the right side of this on this Western blot. And for any of the non-scientists, what a Western blot is it just tells us about what protein levels are present. And so you can look at where it says ER, that's looking at estrogen receptor, and there's no band. And so that tells us that there's no estrogen receptor present. Versus if you look above that and you see T-HER2, which is total HER2, that there is a dark band. And we can use those bands to determine how much protein is present within the cells. And so if we, in this case, this group made cells that were resistant to lapatinib by increasing the doses of lapatinib over time. And you can see that now the cells will, now that the cells are resistant to lapatinib, they respond to ICI and decrease their growth and even have a little bit, it looks like that there might be some death that's occurring as well. And additionally, they have now started to express estrogen receptor. So if you look at that V column under ER, that there's now a band there telling us that estrogen receptor has been turned on. And as expected, ICI is an estrogen receptor degrader, so we see a loss of estrogen receptor in response to ICI treatment. But what about ER-positive cell lines? So the flip of that. So we see HER2, when you block HER2 signaling over time using lapatinib, that now ER is turned on. What about if we block ER over a long time? What happens there? And in this case, this group used the MCF7 cell line, which is an ER-positive, PR-positive, HER2 non-amplified cell line that had a stable expression of aromatase. So I mentioned earlier that the fat is usually where the aromatase is found to convert testosterone into estradiol, and part of that is because epithelial cells tend to not express aromatase, and MCF7 cells do not express aromatase. So what they did here is they caused these cells to express aromatase, allowing for the use of letrozole, which is an aromatase inhibitor. And you can see that over time, the estrogen receptor expression level, this is looking at weeks post treatment with letrozole in a xenograft model, that over time that there's a decrease in the estrogen receptor expression, but that there's an increase in HER2. They then went on to do another study where they used the MCF7 aromatase expressing cells again in a xenograft, and they had a group that they treated with letrozole, and as expected, letrozole initially caused the cells or the tumors to not grow, and then after a certain amount of time, the tumors started to grow. They had a separate group of animals that when they saw that the tumor started to grow, they actually added trastuzumab on, and they saw a decrease in growth in those tumors. So these results suggest that in an estrogen receptor model that we're actually starting to see dependence upon the other pathway again. So now you start to depend on HER2 or the EGFR pathway. Sorry about that. So what about tumors that are HER2 positive and ER positive? We have less data on those. And we are specifically interested in how ER or estrogen receptor is contributing to growth in this context. Like I mentioned earlier, the patients that are treated with either aromatase inhibitors or tamoxifen respond differently whether HER2 is present or not. And so we're interested to see what is ER doing in this context. So what I decided to do was model endocrine resistance in a HER2 positive and ER positive cell line. So there are challenges in modeling endocrine resistance. The first is that Mouse tumors are not dependent on ER. Additionally, that there are a few cell lines that express ER, and that those that do sometimes express but don't actually, again, depend on ER. And media is critically important. So the first thing that's important about media is that phenol red contains an estrogenic contaminant. And so in the bottom left, we're looking at competitive binding affinity, and you can see that estradiol, which is the first line, has a certain binding affinity in MCF7 cells, and then tamoxifen has a slightly lower, but still has a binding affinity. And you can see that phenol red, again, still lower, but it does have a binding affinity. And on the right side, they looked at cell number and saw that with increasing concentrations of phenol red, that there was an increase in cell growth, suggesting that this contaminant, when they added the phenol red back into the media, that there was an increase in growth. And our media, which is the IMM from Gitco, is right there on the scale. So it is affected by the phenol red contaminant that's present. SBS, which is fetal bovine serum, we also add to the media to help the cells to grow. Fetal bovine serum, or FBS, contains a lot of growth factors that are not present in media. But the problem with FBS is that there's a variability in hormone levels. So this graph is looking at different lots of FBS, and you can see across the 8 different lots that there are different steroid hormone levels amongst them. And so if we're looking at how steroid hormones are influencing the way that the cells grow, we need to make sure that our levels are consistent across our experiments, and that they're not confounded by having too high of levels within the FBS. So to model aromatase, what an aromatase inhibitor would be doing to the cells, we use a process called long-term estrogen deprivation. And so we take the cells that are normally grown in 5% FBS IMM that contains the phenol red, we switch them to 5% CCS in a phenol red-free IMM or medium. The problem with this is that the CCS, or the charcoal strip serum, which is, sorry, I forgot to mention that on the last slide, charcoal strip serum strips out a lot of the growth factors and hormones so that you have kind of a baseline level of growth factors. It also has reduced growth factors that may that may signal through HER2, through that EGFR, through HER3, through HER4. And so by stripping, we may be removing some of those important growth factors for HER2. And since we're interested in focusing specifically on the role of estrogen receptor, we want to ensure that we're not affecting the way that HER2 is signaling. And so I'm also developing ICI-resistant or fulvestrant-resistant cell lines by taking the cells that are normally grown in 5% FBS with the phenol red-containing IMM and switching them to, again, still containing the 5% FBS. We're using the phenol red-free IMM, so removing that estrogenic component, and then we're adding increasing doses of ICI or fulvestrant to reduce the amount of estrogen receptor that's present within the cell. And this part of my cell producing is under construction, so I'm not going to be talking about those cells specifically. I'm going to be talking about the LTED models. And so before I get into the LTED models, I want to give you a little bit of background on our parental lines. And so we have 3 cell lines that are HER2 amplified and estrogen receptor positive. And so you can see from the Western blot below that all 3 of the cell lines contain HER2 and estrogen receptor alpha. This figure is split into 2 parts. I'm showing that on the right with the red IMM, that figure will represent for the rest of the presentation that the cells are in their regular base media. And on the left, we have the yellow looking flask, which just shows that the cells are stripped. And what I do, so stripped is actually also a verb. So what I do to strip the cells is I remove the red media and put on clear or yellow media and let that sit, and then change it daily for 3 days before actually using the cells for an experiment. And that just tries to start removing any of the residual hormone that might be present within that media. And so for my LTED lines, which I'll get into in a minute, I'm only focusing on the 361s and the BT474s. The 07530s are under construction still. And one important thing to note is that I'm using two flasks, or I've developed two flasks of each of these cell lines and designated them LTED-A and LTED-B. So if we look at the normal growth within each of these cell lines in response to some of these therapies, you can see that the BT474s are more sensitive to TP or trastuzumab, pertuzumab. So what I'm showing you here is in the black bars are control using the vehicle for each of these that the different drugs are dissolved in. I have two different concentrations of ICI or fulvestrant that I added. Those are the pink bars, and then purple is TP alone at two different concentrations, and then in the blue you see the different combinations. And so again, BTs are more sensitive to trastuzumab and pertuzumab, whereas the 361s are resistant to TP and respond well to ICI. So when making long-term estrogen-deprived cells, there are a few ways to determine whether the cells have actually become resistant. So I'm not increasing concentrations of something to show that a drug is getting to, that we're going to get to a certain concentration, the cells are going to survive in that concentration. We're just removing the red component of the media. And so the two ways that I determine resistance in my LTDs is to monitor the regulation of estrogen-regulated genes. In this case, again, I'm using that PGR progesterone receptor and TFF1, which I mentioned earlier. And then I also measure growth in response to estradiol and ICI. And so first, I'll show you my results from the gene expression. So what I did here was I took the BT474s and I put them through the stripping and then treated the cells with vehicle as my non-treated control, 1 nanomolar of estradiol or 10 nanomolars of estradiol. And you can see that as expected that the progesterone receptor and TFF1 both increased in response to treatment with estrogen. And when I look at the LTED line, so here, again, the lighter colors represent treatment with estradiol, and reds are the LTED-A, and blue is LTED-B. And you can see that there's an increase in the expression of progesterone receptor and TFF1, suggesting that there's not any resistance that's occurring in these cells. So one thing I wanted to check, because all of these results are normalized to their individual vehicles, so the parentals are normalized to their vehicle, the LTDs each are individualized to normalized to theirs, as you can see by the bar for vehicle being 1 across the board. So I wanted to compare the different vehicles to see if there was any effect of long-term estrogen deprivation on the baseline gene expression. And so you can see that there was a slight decrease in progesterone receptor expression, but that estrogen receptor ESR1 is the gene for estrogen receptor, and TFF1 didn't really seem to have much effect. If anything, TFF1 went up a little bit. So then I looked at our 361s, looked at their ER target gene expression, and again, you can see this beautiful pattern where I see an increase. With increased concentrations, there's increased expression of the different genes. However, in this case, I did not see any effect of estrogen on my LTED line, suggesting that these may be starting to be resistant and not dependent upon estrogen receptor signaling. And when I looked again at those baseline levels, I see that not only is progesterone receptor down, it's way down in comparison to the BTs. We also see now that TFF1 and ESR1 or estrogen receptor gene expression, so that's mRNA levels, are decreased. So the next thing I wanted to do was to measure growth in response to estradiol and ICI. And so I took my parental cell lines, and these are in the base media, and I looked at the lighter to darker color are increased concentrations of estrogen. The pink is with vehicle ETOH or ethanol. And then again, increasing concentrations of estrogen, but with 1 micromolar ICI. And then finally, I included TP, which is the 1 microgram per mil trastuzumab and pertuzumab, as kind of a positive control for my BTs to show that there was a decrease in growth. So you can see, as expected in my parental lines, they're responding to both ICI, and then they have a further response, reduction of growth in response to TP. When I looked at my LTD lines, which again are maintained in white media, so they were plated in that as well, there's a slight reduction in the effect of ICI on growth, but they still are responding to ICI with a decrease in growth and as well as a decrease in growth in response to TP. And when I looked at my 361s, as expected, you know, I'm seeing a large decrease in growth in response to ICI and no response to TP. And when I looked at my LTD lines, it looks like they're responding less to ICI treatment. And interestingly enough, we're starting to see that now there's a decrease in growth in response to TP. So it looks like not only are my 361 cells, let me go back for a second, not only are my 361 cells not responding as well to ICI or estradiol, they also are now decreasing in growth in response to TP. And so they seem to be becoming estrogen independent or endocrine resistant, but are they also now responsive to TP? And so downstream targets are used to determine trastuzumab and pertuzumab efficacy. Remember from earlier, these two proteins, HER2 and EGFR, are able to dimerize, and then that dimerization leads to phosphorylation of a variety of proteins, one of which is PI3 kinase, which causes the phosphorylation of AKT. So we can use the phosphorylation of AKT as an indicator that the HER2 or EGFR is functioning, and so when we block that, using TP, we should see a decrease in phospho-AKT and a decrease in phospho-EGFR because the pathway is now inhibited. And so I ran a Western blot to look at the BT474s. In this case, phospho-EGFR. Before I get started, to orient you, on the top I have the first 3 lanes are the parental lines, the next 3 are the LTDA, and the next 3 are LTDB, untreated, treated with TP, or treated with ICI. And so you can see with phospho-EGFR, as expected, TP decreases the amount of phospho-EGFR that's present in all 3 cell lines. When we look at phospho-AKT, we see a similar pattern. So again, TP is decreasing the amount of phospho-AKT that's present. And then, as expected again, when we treat with ICI, you see a reduction or a loss of estrogen receptor because ICI is an estrogen receptor degrader, but estrogen receptor is present in all 3 cell lines. So when I looked at my 361s in phospho-EGFR, I don't really see an effect in the parentals as expected, but I do see now in my LTDs that I'm decreasing the amount of phosphorylation of EGFR in response to TP. I see a similar pattern with phospho-AKT. I think this is even more beautiful and more pronounced. So if you look at the phospho-AKT in the parentals, not only is it lower basally, we also don't see any decrease in response to TP treatment, suggesting that they're resistant to that. But then when you look at the LTDA and LTDB, those bands are almost completely gone in response to TP treatment. And again, you're looking at higher levels of kind of all these parts of the signaling pathway for EGFR signaling. The total EGFR is up, the total phospho-AKT is up. And when we look at estrogen receptor, as expected, we see in the parentals that we lose estrogen receptor in response to ICI, and we also just lose estrogen receptor. So this corroborates our findings in the gene expression where we see that decrease in RNA in response to long-term estrogen deprivation. And here we're seeing, again, the protein level, the estrogen receptor is also decreased. So to summarize the cell models, we see that the BT474 LTED cells maintain estrogen receptor and activity, and they also maintain the TP response. So there doesn't seem to be a great effect of long-term estrogen deprivation on the biology of the BT474s. The MM361s, which again are resistant to TP initially, lose estrogen receptor expression and activity and gain trastuzumab-pertuzumab response. The LTEDs for the 07530s have been largely unsuccessful, but again, I'm continuing to try with those, and the ICI resistant lines are developing. So we can get some answers to see, do those ICI resistant lines match what we see in the LTED, or is there a different phenotype that's occurring when all of those different growth factors are present, but in the absence of estrogen receptor? So our next steps for these cell lines are to use these models to understand the role of estrogen receptor in HER2 positive and ER positive breast cancer. And so what can these preclinical results start to tell us about breast cancer in the clinic? So we went from bedside to bench using some of those clinical trials to determine and start to look at some cell models. How can we take this information and go back to bedside? So we currently use a paradigm where we treat to failure. So we give patients treatments until there's a change. So they get a treatment for an extended period of time until there's a change. So the change means that either the cancer comes back or grows. In some cases, it's an unfortunate side effect. There's a lot of different reasons why we see failure, but they're treated to failure. Clinical trials currently look at targeting both ER and HER2 simultaneously, which definitely shows some promise, but we're proposing that we should potentially be cycling treatments. So kind of taking the biology of this disease and showing that, that the cells in our culture models at least are switching the dependence on their pathway from estrogen receptor to the HER2 pathway and back, potentially. And so can we see, should we be cycling treatments similarly in the clinic? And so at the bench, I'm going to take those LTED cells, the 361s that have become estrogen independent, and put them back into the regular media or the media containing phenol red free and FBS to see what happens to the phenotype there, and then take those cells and make them resistant to trastuzumab and pertuzumab, so increasing, adding increasing concentrations of TP, and try to see if they regain their function. An important question involved in this is what biomarkers should we use to determine the switch? So one of the things I showed you was that a very drastic decrease in phospho-AKT that was not present in the initial cell line. But again, these are cells. So what kind of other, these are cells that represent tumors. And so to have a patient go through multiple tumor biopsies, we have to think about what biomarkers we can use to determine when that switch is happening and how to make that switch. And our future direction, so our lab is moving forward looking at HER2 positive, ER positive breast cancer, and we're trying to determine specifically, again, the ER dynamics and heterogeneity, and see whether they're critical drivers of this biology, and then also the response to HER2 targeted therapy. And with that, I wanted to thank my lab. Dr. Riggins has been a phenomenal mentor and has really given me some scientific freedom to start moving forward with this project. I have a vested interest in HER2 positive breast cancer, and so it's been great to take my background of hormones and move it towards, looking at a double positive situation. Also, all the people at Georgetown, we have some collaborators that are helping us, and of course, always the patient advocates. It is so important to include patient advocates when you're doing research and asking questions about science, because they're the people who will ultimately experience these life-saving drugs that we're working towards. And with that, I will take any questions.
Thank you, Dr. Stiers, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, how do these two different classes of receptor, HER2 and ER, interact?
So I'm going to go back a few slides to show you the, can everybody see that, the HER2 and ER pathways that overlap? So in this case, we see that estrogen receptor can actually has non-genomic effects, and in that kind of case, we see phosphorylation of estrogen receptor by some of these downstream targets, including AKT and MAP kinase, which can phosphorylate the estrogen receptor, allowing for some of those non-genomic effects. And on the opposite way, when estrogen receptor interacts in the nucleus and turns on some of these genes, some of those genes include estrogen receptor itself doesn't cause HER2 or EGFR2, it doesn't influence the protein levels of those, but it interacts with other co-regulators, and so it can, kind of influence it in that way.
Now, our next question, why don't patients adhere to treatments?
So adherence is a problem across a lot of different diseases, but in breast cancer, one of the big things is side effects. So like I mentioned in the presentation, patients will take the drugs for 5 to 10 years. And in that time, if you have a drug that's giving you a side effect, you're taking it every single day, some patients will opt to not take it specifically because of that. Additionally, dosing schedules can be a little bit challenging, so you have to take it every single day. Monitoring treatment, so if you're having, to make sure that patients are actually taking it, it's kind of hard to determine whether patients are or not. And then treatment cost is a big thing. So if their insurance isn't covering it, or even if their insurance is, but there's a high copay, that can kind of deter patients from actually taking the drug.
Now, Dr. Stiers, why is phenol red in media in the first place?
So phenol red is actually an indicator of pH. So we include phenol red because cells, when they're going through respiration and kind of, growing on a dish, we see changes in pH, and those changes can be detrimental to the cell's growth and survival. So we include phenol red as a way to measure, and it turns yellow when the cells become acidic, or the media that the cells are living in become acidic. And so we include phenol red as that indicator.
And do cells express both estrogen receptor and HER2 at the same time?
So this is one thing that we're working on actively in the lab, actually. We are interested to see whether this expression is, if they're happening simultaneously, or if there are cells within the population that some of them express HER2 and some of them express estrogen receptor. And then similarly, we also want to, in the near future, look at tumors. So we have a tumor microarray, or TMA, that we're using to look to see within a tumor if we have a patient who their tumor expresses both estrogen and receptor and HER2, whether they're in the same cells. And we can do that using fluorescence. So we can actually stain for both at the same time, and that will allow us to determine whether it's expressed in both. And we want to move forward kind of depending on what answer that tells us. That kind of will help us to infer what might be happening with the biology with the disease.
Now, Dr. Stiers, it looks like we do have time for one more question, so we'll end with this one. Does resistance occur with the use of HER2-targeted therapies?
So unfortunately it does. It is at a lower rate of what we can tell so far. So one thing I didn't mention, I talked a lot about the use of trastuzumab and pertuzumab, or Herceptin and Perjeta, and a study within the past few years actually showed that the addition of pertuzumab to trastuzumab alone improved survival. And so in that case, we've actually started using standard of care to use both therapies at once. So a lot of our studies looking at resistance look specifically at the use of only trastuzumab or Herceptin alone. And 15 to 24% of patients do have a recurrence, and the recurrence happens within 8 to 11 years. So that's like the percentage of patients that have recurrence at that time. These drugs have only been around since the 90s, so we don't have as much data as we do on the estrogen-targeted therapies, but unfortunately it does seem to also happen there.
Thank you, Dr. Stiers. Do you have any final comments for our audience?
I don't think I do. Thank you all for listening. I really appreciate everyone being here, and feel free to tweet at me if you have questions. I will post the article that this figure of the HER2 and ER target pathways. I'll tweet that out to the people who are interested in looking to see. I also have a publication about adherence. I'll tweet that out too. So follow me along on Twitter.
Thank you again, Dr. Stiers, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand through June of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.